keeping up with our motto, let learning be a joy and teaching a pleasure. Here we are with a remote teaching and learning process to bridge the gap. Happy learning, students.
Hello students. Today we are going to see the chapter from Science and Technology part 2. Chapter number 2, that is Life Processes in Living Organisms part 1. Students, we have already covered first 3 parts of this topic. In these 3 parts we have covered the topics like living organisms and life process, living organisms and energy production, some nutrients and energy efficiency, cell division, and mitosis. These are the topics which were covered in the first 3 parts. In today's part, we are going to cover the last topic, that is meiosis. Before starting with the part of meiosis, let us understand one term called as homologous chromosomes. In science, the word homo means same. So homologous chromosomes, these are the chromosome pairs that has same length, same centromere position and same genetic sequences. Such chromosomes which have same length, same centromere position and same genetic sequence are called as homologous chromosomes. In the diagram, you can see a structure of the chromosomes. This chromosome is called as paternal chromosomes. That means from father. Whereas the next structure is called as maternal chromosomes, that is from the mother. This paternal chromosomes and maternal chromosomes, they together form a pair of homologous chromosomes. This homologous chromosomes, we are going to see it further in this part. Now, coming to the actual part of meiosis. Meiosis is a process which occurs in germ cells. Germ cells are the cells which are called as reproductive cells, if you recall, which we have done it in the earlier part, that is part 2. In males, it is called as sperm cells, whereas in case of females, it is called as egg cells. So this meiosis is been carried out only in germ cells. Now this meiosis is carried out in 2 steps. First step is meiosis 1. In meiosis 1, there are different phases, that is prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis. These are the stages which are carried out in meiosis 1. Once the stage of cytokinesis come, it moves on to the next stage called as meiosis 2. And this meiosis 2 is further carried out in 5 stages. That is prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. This is the last stage in cytokinesis where your meiosis 2 gets completed. Starting out with the very first step, that is meiosis 1. In meiosis 1, the first step is called as prophase 1. This prophase 1 is carried out in different stages. There are 5 stages where this prophase 1 is carried out. The first stage is called as leptotin. Second stage is called as zygotin. The third stage is called as pachitin. The fourth stage is called as diplotin, and the last stage is called as dikinesis. So your prophase 1 is carried out in 5 stages, that is leptotin, zygotin, pachitin, diplotin, and dikinesis. Starting with the first stage, that is leptotin. In leptotin, the chromosomes condenses. That is, the condensation of the chromosomes starts in this stage and they become thick and visible. Once they starts becoming visible, they move on to the next stage called as zygotin. In zygotin, as you can observe it in the diagram, the chromosomes starts forming the pair. So, in zygotin, the homologous chromosomes lines up with pairs. That is, they start forming the pairs. Once the pairs are formed, it comes to the next stage called as pachitin. In pachitin is a stage, very important stage, where crossing over takes place or mixing of two chromosomes occurs between homologous pairs. Once the mixing takes place, the next stage, that is diplotin, comes. Here, in case of this diplotin, the crossing over chromosomes start moving away from each other. That is, repulsion of homologous chromosomes starts here. And the repulsion of the homologous chromosomes, which is started in diplotin, continues in dikinesis, and in dikinesis, if you observe it properly, the two homologous chromosomes starts separating. That is, finally they are separated as homologous chromosomes. So these are the 5 stages of prophase 1. The first is called as leptotin, where the condensation starts. Second stage is called as zygotin, where the pairing takes place. Third stage is called as pachitin, where the crossing over takes place. Fourth stage is called as diplotin, where the repulsion of the chromosomes takes place. And the last stage is called as dikinesis, where the separation of the homologous chromosomes is carried out. Once this separation is carried out, it is ready for entering into the next phase, that is called as metaphase 1. In metaphase, as if you recall what we have done it in the mitosis. In any metaphase, the normally thing which is carried out is the chromosomes arrange themselves in the equatorial plane, as it is seen in the diagram. So here also the same thing is carried out, that is, the crossing over pairs are arranged along the equatorial plane of cell. That is how the metaphase 1 is completed. Once the metaphase 1 gets completed, it gets ready for the next phase called as anaphase 1. In anaphase, if you recall in mitosis, the common thing which happens in anaphase is the sister chromatids, or the separation of the chromosomes takes place. So same thing happens in this diagram. The homologous chromosomes are separated and pulled at the opposite poles. So the separation has been carried out here, which is continued further in telophase 1. where the chromosomes get separated and nuclei begin to reform. Once the nucleus gets formed, two new daughter nuclei are formed inside the cell. This is how your telophase 1 is completed. Once it gets completed, it moves on to the next stage. Till telophase 1, the nucleus has got divided. Now it's the part for cytoplasm to get divided. So that is carried out in the next stage called as cytokinesis. That is, in cytokinesis, the cytoplasm splits and two new cells are formed separately. So here the cytokinesis stage is completed and your meiosis first stage is completed. So in meiosis stage 1, that is meiosis part 1, we got two different daughter cells. These two daughter cells which are formed in the meiosis 1 are used for the next phase, that is meiosis 2, which is again carried out in the same phases. The first phase, that is prophase 2. In prophase 2, the two daughter cells which are formed earlier, in this case it starts condense and appears thick. Then it moves on to the next stage, that is metaphase 2. The spindle fibers which begins to form in the prophase 2. This spindle fibers helps the chromosomes to arrange themselves in the equatorial plane. So the chromosomes are arranged in the equatorial plane. These chromosomes which are arranged in the equatorial plane are used in the next stage called as Anaphase 2. So in Anaphase 2, as we have seen earlier, the separation takes place. The sister chromatid are pulled by spindle fibers. And once they are pulled by spindle fibers, it moves on to the next stage, that is telophase 2, where in the spindle fibers which are pulling the sister chromatids, the cell starts to divide into four daughter cells. That is, four daughter cells are formed in telophase 2, which is continued into the last stage, that is cytokinesis. In this cytokinesis, the daughter cells which are formed in the earlier stage, the chromosome splits by forming the notch in the middle of the cell, and four new haploid daughter cells are formed. So the two daughter cells which are formed in the meiosis 1, that is further continued, and we are getting four daughter cells in this stage. These cells become sperm cells in case of males and egg cells in case of females. So that is how your meiosis 2 is completed. Thank you, dear students.